Hello, and thank you for coming to my poster titled High-Throughput Chemical Safety Screening Using Targeted RNA-seq. My name is Logan Everett, and I am a bioinformatics scientist at the United States Environmental Protection Agency in the Center for Computational Toxicology and Exposure. First, a quick disclaimer. Please note that the views expressed are those of the presenter and do not necessarily reflect the views or policies of the U.S. Environmental Protection Agency. Standard chemical safety testing approaches are time and resource intensive and have led to large knowledge gaps in the potential risks posed by thousands of chemicals. EPA has recently proposed a tiered testing strategy to accelerate the pace of chemical safety screening. You can read more about the overall roadmap for the tiered testing strategy in reference 1, but the main thing to know here is that the first tier in this strategy will utilize methods that are both high throughput and provide broad biological coverage. One such method is targeted RNA-seq, which we refer to as high throughput transcriptomics, or HTTR. Our center at EPA recently published results from a successful pilot study using HTTR for Tier 1 chemical safety screening. See reference 3 for more details on both the laboratory and bioinformatic methods. We have subsequently scaled this methodology up to screen over 1,200 chemicals in 3 distinct cell lines. This tidal wave of data required the development of a reliable and automated processing pipeline, the results of which are highlighted in this poster. Figure 1 diagrams the design and analysis of our large-scale screens. The left side shows the general experimental design using 384-well plates to expose cultured cells to multiple concentrations of multiple chemicals. The right side of the figure shows an overview of the bioinformatic pipeline to rapidly align, store, and model the resulting data. Figure 2 shows the results of our QC metrics based on the alignment and subsequent probe counts from over 100,000 total HTTR samples. The upper panels show that the majority of samples have sufficient read depth, which is generally much lower than what is needed for traditional RNA-seq, and that the majority of reads uniquely align to known probe sequences. Additional QC metrics based on the distribution of counts across probes are shown in the lower panels. These metrics change for different cell types, so a distinct threshold was set for each case. Figure 3 shows the overall QC failure rates by sample and cell type. In addition to the bioinformatic QC checks from the previous figure, represented here in blue, additional laboratory data was also used to exclude individual HTTR profiles from further analysis, including errors in the acoustic dispensing system and issues with acute cytotoxicity in response to certain chemical treatments. Combining all of these QC filters only removes on the order of 1 to 3% of individual HTTR profiles out of tens of thousands of samples per cell type as tabulated on the bottom here. This demonstrates the ability to reliably scale our workflow to studies encompassing thousands of chemicals and samples. The upper left portion of Figure 4 shows that differential expression patterns are largely concentration dependent across the tested chemicals. The number of differentially expressed genes responding to each concentration of each chemical were determined using DE-seq2. The box plots here show the number of accumulated DEGs per chemical at each concentration and in each cell type. The majority of chemicals do not significantly perturb any expression levels at the lowest concentration, but perturb the expression of an increasing number of genes as that concentration increases. The right side of Figure 4 shows that differential expression patterns are highly reproducible, especially when converted to signature scores. Log2 fold change responses were estimated using DE-seq2 for 3 standard reference chemical treatments included at a single concentration throughout the MCF7 screen. As a result, we estimated 37 independent fold change profiles for each of these 3 chemicals, and each of the profile is based on 3 replicate treatments, each from 3 test plates. Correlations for each fold change profile were computed against the median profile for that chemical, and the distribution of these correlation coefficients are shown in red here. We also computed signature scores using the single sample GSEA method and a gene set catalog described in our previous paper, see reference 3 on the poster, and computed correlation distributions as was done for the fold change profiles. Replicate correlations based on signature scores were generally higher than those based on fold change profiles. This suggests that signature scores improve signal to noise in these high throughput datasets and therefore may be more reliable for concentration response modeling. In summary, the vast majority of HTTR samples, greater than 98% overall, pass multiple layers of rigorous QC tests even when scaling our screening platform to the level where tens of thousands of targeted RNA-seq samples are generated per study. The reference chemical treatments included throughout our large screens demonstrate the reproducibility of differential expression profiles. These differential expression patterns are generated at 8 separate concentrations for the test chemicals, spanning several orders of magnitude, and can therefore be mined to infer potency as well as mechanism of action for each chemical. Thank you again for coming to my poster. I'm happy to take further questions using the conference chat features, or feel free to contact me at the email address shown here.